Today we will discuss about how this MET helps in the activation of CXCL8 by converting the dimer CXCL8 into the monomers. In the previous video we have discussed that METs helps in the metastasis of the pancreatic tumor cells by upregulating the ICAM on the pancreatic tumor cells, and those ICAM then interact with the endothelial cells, and thereby those endothelial cells helps in the metastasis of the pancreatic tumor cells across the different organs, especially in the liver. So here they have discussed the results and try to find out how this MET uh, helps this cytokine CXCL8 that thereby helps in the metastasis of the pancreatic tumor cells. So the hypothesis that they developed is that MET helps in the activation of the CXCL8 and thereby those CXCL8 can leads to the high ICAM production on the high ICAM expression on the pancreatic tumor cells. So in order to identify whether the CXCS that are formed are activated or not, they developed a process where they first take the cultured medium from the co-culture of the pancreatic tumor cells and the macrophages, and do the size exclusion chromatography to distinguish to find out these CXCL8 dimers and CXCL8 monomers using the ELISA detections. They are able to check whether this CXCL8 is in the dimer form or in the monomer form. In the monomer forms is found to be more biologically active. After they have developed this technique to find out the activated CXCL8, they checked the CXCL8 in the pancreatic cancer cells which are cultured with the cultured medium of tumor cells, the macrophages that contains the cytokines. And same things they did with the ASPC. And what they have find out is that when they use the culture medium from the pancreatic cancer cells that have overexpressed MLKL and the macrophages, they see that increase in the production of this activated ICAM, and same things they observed for the ASPC cell lines. And once they find out that this activated ICAMs are the results of the co-cultured medium that they have obtained from the co-culturing macrophages and the tumor cells with high MLKL overexpression. They decided to check whether it has any impact with these MET inhibitors or not, and they use different kind of MET inhibitors. And when they use the general MET inhibitors, they find out that this activated CXCL8 is reduced, and for the MLKL inhibitors also it is reduced. When they check for the individual inhibitors, what they find out that it is reduced for DNAs, it is reduced for this MMP12, and also it has shown to be reduced for DNase I. So DNase inhibitors they have used because the process by which this CXCL get activated is that it interact with the DNA scaffold. It interact with the DNA scaffold of the matrix metalloprotease that then cleaves the CXCL8 dimers into the more activated form monomer CXCL8 of 77 amino acids. So what they find out for MMP12 and DNase I, they see the significant reduction in the activated. CXCL8 compared to the total. However, they see that small reductions in the case of this MMP2 and MMP9, and similar results they have observed for this co-culturing of this ASPC1 co-culturing using co-culture medium from the tumors and macrophages in the ASPC1 cell lines. That is, the reduction is of the activated CXCL8 is high in the case of when they use MMP12 and DNase. Here they use MMP12 and METI, and also here they do not use DNase I. but the reductions is also seen in the case of MMP2 and MMP9. And after this, they check their results using the confocal results of this MMP2, MMP9, and MMP12, and they can see that in the case of MMP2 and MMP9, the overlap of this MMP2 signals and CX MMP2 signals and CXCL8 signals are low. That is shown on the right hand side quantifications. For MMP2 and MMP9, the overlapping is low, whereas for the case of this MMP12, this overlapping is high, and it is visible also, indicating that MMP12 has a higher role. MMP12 has a higher role towards the activation of this CXCL8, towards the activation of this CXCL8, and after validating that is the MMP12 that plays a role towards the activation of CXCL, they check the MMP cleavage site, and the cleavage sites at this region is MMP cleavage site in the case of CXCL8 is overlapping MMP cleavage site in the MMP12 cleavage site is overlapping with the CXCL8 sequence from 23 to 28. 23 to 28 sequence of the CXCL8 is overlapped with the MMP cleavage site, indicating that is the MMP12 that helps in the activation of CXCL8. And these things they have validated using the Western blot also, where they have seen that when they use MMP12 inhibitors, there are less CXCL8 monomers formed and the more CXCL8 dimers formed compared to the other inhibitors. So all this indicates that it's when the co-culturing, when the interaction of the tumor cells with the macrophages occurs, it will cause necroptosis of the tumor cells that leads to the release of the CXCL8, and this CXCL8 will further lead to the upregulations of this CXCL8 through this MMP12, and that in turn helps in the metastasis or upregulations of this ICAM on the tumor cell surface that can interact with the endothelial cells, and thereby endothelial cells helps in the metastasis of the tumor cells across the different organs. So I hope you understood the story so far that is mentioned in this paper. If you need to understand more about this paper's results or the results of this experiment or the previous experiments, you can either go through the reference that is included in the description, or you can refer the previous videos that are related to this paper. So that's all for this video. So thank you for your time.